Hello, my name is Nursyahira Nafisha binti Abdul Razak and my supervisor is Dr. Fazlina Nadia binti MD Akhir. So today, I would like to present my final year project entitled Antibiotic Resistance of Lactic Acid Bacteria Isolated from Dairy Product in Malaysia. Before I go further, this slide shows all the contents that I will be present and discuss in this presentation. Generally, antibiotic are medicines used to prevent and treat the bacteria infection. Antibiotic resistance is rising to dangerously high level in all parts of the world. New resistant mechanisms are emerging and spread globally, threatening the ability to treat common infectious diseases such as pneumonia or tuberculosis become harder and sometimes impossible to treat as antibiotics become less effective. This fact can be linked to a global study on antibiotic resistance which over 700,000 people die each year, with a projected increase to 10 million die by 2050. In addition, the usage of the antibiotic increased by 36% between 2000 until 2010. This shows that without standard treatment guidelines, antibiotic will be overused by the public. Without urgent action, we are heading for a post-antibiotic era in which common infection and minor injury can once again kill. Antibiotic resistance involves a variety of processes, including the presence of resistance genes that allow the active antibiotic molecule to be inactivated directly, as well as the loss of antibiotic susceptibility caused by changing the target site or lowering the antibiotic absorption. Therefore, antibiotics are ineffective, and resistant microorganisms can survive and transfer their resistant machinery to other microorganisms and become a public health threat. Let's move on to the objective of the study. Firstly, to identify the dominant lactic acid bacteria that present in dairy product in Malaysia market. Secondly, to investigate the antibiotic resistant profile of most commonly used lactic acid bacteria starter isolated from dairy product in Malaysia market. Third one, to characterize the corresponding resistance gene involved in antibiotic resistance. Next is methodology. For the identifications of lactic acid bacteria from dairy product and its growth state, lactic acid bacteria strain were isolated from the commercial dairy product. So briefly, the dairy sample were homogenized and then serially diluted in the sterile saline water. For the isolations of the bacteria, diluted sample were placed on LB broth and incubated anaerobically in anaerobic chamber at 37 degrees Celsius for 48 hours. So during the isolation, a 300 mg of yogurt sample will be used and diluted into 900 mg of sterile saline water by using the autoclave Eppendorf tube. Next, for the lactic acid bacteria recognition, PCR-based assays were adopted for the identifications of lactobacillus and streptococcus. So all the isolates were tested for gram staining, catalase and oxidase activity, cell morphology and spore formation. So the bacteria strain were tested for the productions of acids from carbohydrate and related compound by using the analytical profile index STRAPS kit. So the results were collected after incubation for 24 hours and 48 hours at 37 degrees Celsius and analyzed for the species identification. For the antibiotic susceptibility testing, this method was determined by using a test diffusion method. Then a single colony of the lactic acid bacteria was inoculated into the respective broth and incubated at 37 degrees Celsius for 24 hours. All the antibiotic tests containing vancomycin, neomycin, gentamicin, and streptomycin. So the zone of growth inhibition around each of the antibiotic discs were measured, including the disc diameter, and classified as the susceptible, intermediate, or resistant respectively. For the PCR detections of the gene for antibiotic resistance, the genomic DNA of lactic acid bacteria strain was extracted using the Presto Mini gDNA bacteria kits. So all the genes responsible for the resistance to vancomycin, gentamicin, neomycin, streptomycin, then investigate by using the PCR with the primer. And the PCR amplifications was performed using the thermal cycle. So this table shows the PCR primer used for the detections of antibiotic resistance gene in lactic acid bacteria strain. Next is result and discussion. Studies conducted by Wong et al. Analyzed that resistant profile to different types of antibiotics, including two types. First is inhibitors of cell wall synthesis, such as the glycopeptide for vancomycin. Secondly, inhibitors of bacterial synthesis on the TTS ribosomal subunit, such as the aminoglycoside for neomycin, gentamicin, and streptomycin, and their corresponding resistant gene. Regarding to their study, the presence of the antibiotic resistance was identified in 8 out of 9 lactic acid bacteria strain isolated from the fermented dairy product, and the resistance gene were identified in 6 resistant strains. Overall result, they analyzed the resistance strain to neomycin, gentamicin, streptomycin, and vancomycin were found in 77.8% for neomycin, 66.7% for gentamicin, 44.4% for streptomycin, and 11.1% for vancomycin. These data were in accordance with other reported a high percentage of lactic acid bacteria strain that were resistant to aminoglycoside. Membrane impermeability has been regarded as the main mechanism behind the resistance of lactic acid bacteria to aminoglycoside because most of species of this genus lack the cytochrome-mediated electron transport that can mediate the antibiotic uptake. Besides that, the study carried out by Zhou et al. They isolated 266 lactic acid bacteria strain from 28 Dahi sample with Streptococcus, which is 50%, which is being the most dominant genus, followed by the Lactobacillus 27% and Lactococcus 5%. This supported by the studies carried out by the Ozdemir and Bas-Bulbul, the presence of the multi-resistant strain as well as the PCR positive result for many resistance genes indicated that Streptococcus was the most risky genera in their study in terms of the antibiotic resistant trait transfer. Similarly, the study carried out by Wong et al., the sequencing result for the APH and AADA2 were analyzed by BLAST, and identification to the gene in GenBank is described in Table 3. Sequence for amplicons of APH show that 100% similarity to the gene encoding neomycin phosphotransferase 2 in gene replacement vector TEXKM4. Sequence of the AADA2 amplicons were validated to be 99% identical to the gene responsible for the tRNA adenosine deaminase in Lactococcus. So in the current investigation, some resistant strain lack related resistance gene. AADB was not found in any of the gentamicin resistant strain, for example, so this outcome could be explained by a variety of variables, suggest the mutation may be blamed for the evolutions of the resistance in certain strain. As the conclusion, antibiotic resistance is a complex issue that necessitates a broad multidisciplinary approach to predict and avoid negative public health consequences throughout the entire food producing chain. There have been few systematic studies on developed antibiotic resistance in lactic acid bacteria of food origin. Most data are on Streptococcus with a small number of reports on Lactobacillus. In the United States, after evaluating general elements of the safety taxonomy antibiotic resistance, and the historical context of food safety, the Food and Drug Association categorized microorganisms with the general consumption as safe, which is grass. The spread of antibiotic resistance gene put the grass category in a different light, particularly for bacteria that have the mobile gene of transfer, such as lactobacillus, because there are still no guidelines in the United States that address the types of resistance in food processing microorganisms. The EU Commission monitors the safety of lactic acid bacteria used as a starter or probiotic culture in Europe through the European Food Safety Authority (EFSA), which has established rules for giving qualified presumption of safety quality to organisms since 2003. This kind of guideline should be strictly implemented by the government of Malaysia to ensure that dairy product that's supplied to public is safe to consume. That's all from me. Thank you.